Thank you so much for having me, and thank you, Cameron, for delivering this great talk. It was great to hear that, and Cameron's one of the probably most experienced data sharing people in neuroimaging I know. So it was great to have you involved in the project and delivering the speech. Okay, well, welcome everyone. I hope you are all well awake and prepared for talking about sharing statistical maps. Okay, so basically my motto is every voxel counts. Or in other words, there are different types of data sharing. And if you think about it, what is data? It's basically some machine readable piece of information. So even if you publish a paper and post the coordinates of activation in that paper, that's already a form of data sharing. Because those coordinates can be extracted from the paper and something useful can be done with this. And many people have been doing meta-analysis using those things. But if you want to go a step further, you can attempt to use all of the information in your contrast map. So all of the information in your statistical map, not just the peaks and the locations of the peaks. And what I'm going to try to argue is that there's a lot of information there, and sharing this information can be not only made easy, but also rewarding for the person doing the sharing. Okay, so who in this room has in the past done any sort of statistical contrast and done basically brain mapping? Yay, yay. This is what I was expecting when I attended the Organization of Human Brain Mapping. So good, I'm at the right conference, so far so good. But if you think about what we are sharing after those hours upon hours and thousands of dollars spent in a neuroimaging experiment, what do you get out of it? Well, you get a paper, which is a synthesis of knowledge. And in terms of machine readable data, if you're lucky, you get that table of coordinates. So that sort of seems like throwing money away if you kind of see the compression of the data from the like raw nifties into this little table in the paper. So the question is, can we push this a little bit further and get a little bit more data? And the reason why we didn't do that before is because we are sort of trapped in this format of delivering knowledge as a paper. And a paper used to be a physical thing only, and now it's more of a PDF, but it's still formatted like a physical thing that you have to print out, and that's the only thing that counts at the end of your study. So the question is whether we can do something, and our attempt to help with this was to make things simpler. So we want to minimize the cost of data sharing. So we want to make it as easy as it can be. Basically, the investment of time into making a cup of coffee would be as hard as sharing your statistical maps. And the trade-off with data sharing is basically not only having the data itself, but having a meaningful annotation of the data. So if I only have a bunch of bytes, not knowing where those bytes are coming from or numbers, I won't be able to do anything. But here we sort of taken a shortcut and assume that the best description of your experiment is the paper you have written about this experiment. So as long as we have the DOI of the paper corresponding to the statistical map, a lot of really interesting application can be derived from it. We also want to streamline logging processes. So basically, you can go on the website, log in with your Google account or Facebook, although I did hear recently that Facebook is not very cool among young people anymore, which makes me feel a bit old. Okay, nonetheless, and then you can create a collection of your maps. One collection corresponds to one paper, and you can add your maps, upload your maps, and benefit from various tools and analysis mechanisms that we've built into the platform. And this is the carrot. This is the thing that you as a researcher sharing your data can benefit from. So first of all, we have visualization. And this in itself can be very useful because you can upload your map and you can send a link to your collaborator or maybe to your supervisor who is at a very important business meeting in Hawaii. And they can open the email and they don't have to open a nifty viewer, download the thing. And like, there are some PIs that actually don't deal with data anymore. But they can just click the link, open the website, and everything is in the browser. We also have very, very fancy three-dimensional visualization coming from Jack Gallant's lab. Anyone remembers those fancy decoding brains? Yes, you can get exactly the same visualization for any of your maps if you upload it to NeuroVolt. We also have cognitive decoding. So who heard about Neurosynth? It's great. So Neurosynth is a project run by Talia Arconi that crawls many papers out there and tries to synthesize neuroimaging knowledge in form of topical maps and reverse inference maps. And the platform he built provides a means to do cognitive decoding. So in other words, you provide a pattern of activation in the form of unthresholded statistical map, and that tool tells you what is the similarity of this pattern to topic extracted from the literature. So that can help you to quantify your interpretation of the results you have. Again, click of a button. Other thing that we start benefiting from more recently is that we have similarity search. So you can upload your own map and run a search across all of NeuroVolt to find other patterns, other maps in the database that show the same pattern. And this way you can discover other papers and other publications that might be of context to your discovery. And we can also drill down to it and see exactly what is the correspondence and what is the correlation between those maps and how does it distribute among different parts of the brain. And then we also have gene decoding. So who here heard about the Allen Brain Institute? Excellent, excellent. It's more people in Neurosynth by 2. I counted you. So Allen Brain Institute a while ago did this project where they analyzed 6 cadaver brains, and they took tiny samples of the tissue in different locations, and you can see those locations. Is there a pointer here? Use the mouse. Oh, yes, I can. Yes, very bright. Okay, can you see the dots on these plots? Yes, so each dot is a different location where the tissue was sampled, and they took those tissue samples in those 6 different cadavers, and they've estimated expression of 20,000 different genes. So what we did, we took this data and built in a tool that allows you to see whether the pattern of activation, or basically any brain pattern that we upload to NeuroVolt, whether it relates to gene expression pattern. And that you get with a click of a button. And you get corrected p-values, effect sizes, etc., etc. We validated this using PET maps on neurotransmitters, and we know it works to a certain level of specificity. So that's another way for you to very quickly extend the context of your finding now not only to other neuroimaging studies, but to gene expression data. Okay, and then we very recently added support for free-surfer, FS average surface data. So if you don't work in volumetric space, this is also supported. All right, and last but not least, you can use NeuroVolt to perform a power analysis. So before you start running a study, you can find a study that has a similar contrast in NeuroVolt database and use that contrast map to estimate how many subjects you will need to acquire in your new study. And this is with collaboration with Jaka Durnes, who actually is not a collaboration, she did all the work. We just added a button. So Jaka should take all the credit for this. Basically, there's a button on NeuroVolt where you can go from any individual map, and that will take you to NeuroPower tools that Jeanette already mentioned for doing map-based power analysis. All right, so, that works. Okay, last year, I did a demo, which was super awkward because I had to stand there and shout, and it was weird. So this year, I
recorded a video showing you how to upload the map. So we just logged in using your Google account, and now we are creating a collection. We can give it a name and different options here. We can make it private and make it public only when the paper goes out, for example. And then we can upload our maps. And this is easy as pointing to a folder with a bunch of Nifty files. And when the Nifty files transfer, we get all of our data here. We got beautiful visualizations, and we can fix the metadata. So for example, here, we're going to ask you to tell us what level of analysis this is, whether it's single subject or group level, meta-analysis, as well as how many subjects were used in the study. And you can enter it for one map and then basically copy the same value everywhere else, so that doesn't really take that much time. We also ask you what cognitive paradigm this is. And all of these pieces of information will make your data more discoverable and valuable for people doing meta-analysis. Okay, and when this is done, you can go and start taking advantage of all sorts of different tools that we built and I just talked about. So, for example, we can do cognitive decoding, which takes us to the Neurosynth website. And here on the right, you're going to see all of the different topic maps that correlate spatially with the map that you've just uploaded. You can overlay them and basically get more insight into your finding. We can also do gene expression decoding. No, wait, sorry, similarity. See, I don't remember. So we can also search using this map. And this is very similar to Google image search. We basically look for all of the maps in the database that have the same pattern. And because the database now, it's quite big, you can actually find some interesting studies there and relate to them. And you can see how strong this correlation is. And this way, basically extend the context of your finding. And you can limit this correlation to different subparts of the brain to basically see how robust it is to different outliers. And then we can do gene expression decoding. So this takes 2-3 seconds because we have to go through 20,000 different genes. And when it's done, you're going to see a table with different gene names. And now this table also includes links to NIH ontologies of those genes. And you can search for a particular gene, so you can test specific hypotheses. You can basically say, like, I want to see how this map relates to this particular serotonin receptor. And you can do that using this gene expression tool. And all of this is in the browser. You don't have to install anything. It's just there. And finally, you can do power analysis, and that takes you to NeuroPower, and then you have to fill a few more fields, and eventually you're gonna get an estimate of how many subjects you need to acquire to reach the same level of power as the study you started from. Okay, so this is how it works. We talked already about different benefits and private collections. So that's kind of important to stress out because a lot of people have this concern of not willing to expose preliminary results. So which is perfectly fine. And we support this case. So you can put your maps out there as a private collection. And that means that there is this secret URL that you can share with anyone, but it's not indexed by Google, and it's really, really hard to guess. And you can put that link in your manuscript as well, but we're not going to index your maps in any of the main sites of NeuroVault. So that's a private collection, basically. And when your paper gets published, you can turn it into a public collection, and you're not going to get scooped if this is your concern. The other thing that's worth talking about is putting this data out there is relatively cheap in terms of effort, but extends the impact and exposure of your research. And this is not only because, and we're going to show you some examples of that later, other researchers can basically take your maps and use them as, for example, seed ROIs or do quantitative comparisons by doing correlation analysis or meta-analysis. But also we have very good Google indexing. So basically this presence on the web will give your paper even more exposure. Okay, so we did some validation with this a couple of years ago, where we took maps in NeuroVolt and we tried to ask the question, how much do we gain from doing meta-analysis using unthresholded statistical maps versus coordinates. So in the top row, you see peak coordinates from Neurosynth. Basically across many studies. There was no particular narrow filter. It's basically everything that is there at that given time. And the middle row are thresholded maps, which sort of simulates peak coordinates. So we only get the top of the iceberg. But if we use unthresholded maps, because those two, like top and the middle row, look fairly similar, but when we look at the middle row, which are unthresholded map, we start seeing this negative deactivation. And who can tell me what is it? That blue thing? It's the default mode network. Yes, the mystery of the human brain. Yes, so this is the default mode network, basically task deactivation network, which we basically see when we include unthresholded maps, because that's the signal below the positive threshold. And this is just an example on a very broad and specific meta-analysis. We also did some more specific meta-analysis, and basically using unthresholded maps gives you more power when you do this sort of stuff. We also have support for programmatic access of NeuroVolt, and people have been building apps based on NeuroVolt and interacting with NeuroVolt. So you can access an API that allows you not only to browse, filter, download, but also upload maps automatically. NeuroVolt also supports this standard called NIDM results, which is a very rich semantically standard for describing all different aspects of inference and model fitting. And through this standard, we have a button in SPM 12, which is the... Wait, what? What? Why not?
Your version maybe, but no downloader version doesn't have that yet.
But I have been lied to. My follow-up sentence was, "This is the biggest achievement of my career." So thank you for that. I just gonna go cry in the corner now. Okay, so I was told by Guillaume Flodin that this is in the released SPM 12. When you get the results and that fantastic table with peak coordinates that a lot of people stared at with fear, you can right-click on it, and then there is the upload to NeuroVolt menu. But if it isn't there, I will have to have a conversation with Guillaume. Okay. So moving on, we start seeing reuses of NeuroVolt. And because it's sort of a snowball effect and it takes a while to have enough maps to do really big meta-analysis, the first reuses of NeuroVolt were very simple, where basically someone said, we have compared our results with the maps found in NeuroVolt from this other study, or we did seed-based connectivity using the ROIs derived from the maps found in NeuroVolt. We also did some validation of the image search, kind of basically trying to answer the question whether thresholding or thresholded or unthresholded maps, how do they compare in terms of trying to figure out which tasks correspond to others. And lo and behold, it turns out that unthresholded maps are better at searching. But most recently, there was an interesting big meta-analysis from Lucina Udin Group and Thomas Yeo, which took maps from HCP and NeuroVolt and derived the different components underlying the structure of the the brain cognition relationships. And I recommend you to have a look at this paper. And they've used a rather small subset of NeuroVol database because it was a while ago when they started the study, but it's constantly growing. So you can basically redo this analysis right now with much more data. And we hope that there will be more and more stuff like that, which for people contributing to NeuroVol means that there will be more citation for their papers because they all contribute to this meta-analysis. Okay, but NeuroVol is not the only database for results of MR studies. The honorable mentions goes to ANIMA, which is the database dedicated purely to meta-analysis results, run from Ulich Group and Simon Eitkopf, made by Andrew Wright. There is also a database dedicated to connector matrices, and that's the USC multimodal connectivity database. And finally, there's an HCP database for HCP-styled studies, and that is run by David Van Essen. And the last one, is tightly connected to that ecosystem of HCP tools and HCP acquisitions. Worth checking out nonetheless. Okay, so going back to our green triangle of different types of data sharing, we talked a little bit about tables of coordinates and then unthreshold statistical maps. But if I only whetted your appetite for sharing data, there are other ways of doing that, and many people are in this room are involved in data sharing initiatives. For example, Cameron will gladly accept in the FCP in the database many different types of MR data. So if you want to go beyond statistical maps, you can also share raw data and even and increase even more the impact of your research. And with that, now I want to thank different people involved in this project, and I'm happy to take any questions together with all the other speakers.
Thank you, Chris. Okay, so we still have a bit of time. If you have any questions on the usage of the tool or even on the previous talks, feel free to ask whatever questions you want. And then remember that also we're going to demo a lot of these tools during the week in the open science space and the hack room. Zero question?
Don't be shy. I guess everyone is super excited for the Talairak lecture.
That must be the case.
And not at all confused and lost.
Well, that's because NeuroVolt and BIDS are so easy.
Yeah, right. But yes, so if you later come up with any questions, I'm very happy to talk to all and any of you. So
Most of us will be many times during the week will be on the open science space. So please feel free to drop by if you have any questions or you want to check something on your data and you have your laptop with you, whatever.
I see a raising hand there.
Well, I was wondering if you could tell people where the open science space is, just in case they don't know.
Which room is open science space in?
Right, 210, and also basically when you just come up from the stairs, that's it. All that space is open science space.
So room 210.
Plus the whole foyer in front of it.
Yeah, exactly. The second floor foyer.
The whole foyer, whatever foyer is.
It's an open space where you sit and talk with people.
That thing.
All right, thank you everybody.